Good afternoon, everyone. Welcome to the H3D Foundation webinar series. Our today's speaker is Dr. Andre Horatscheck. Andre is Senior Investigator at the H3D Center and is a malaria project lead since April 2018. As we all know that it was World Malaria Day on 25th April, and it is observed to spread awareness of the global efforts to control and ultimately eradicate malaria. So this webinar is an attempt to highlight the need for continued investment and sustained research commitment for malaria prevention and control. So before we start, the common rules that we will have a Q&A session after the talk. So questions can be posted in the chat or can be kept for the end of the talk. Today's topic is malaria resistance is futile or not. And now I would like to request Dr. Andre to share his work with us. Over to you, Dr. Andre.
Yeah, thank you very much. As Shruti already mentioned, we had World Malaria Day yesterday. And so what I want to do is I want to highlight some of the work that is being done and more show a broad overview about different approaches that are being conducted across the world to eliminate malaria. You may realize, or some of you who know Star Trek, you may recognize resistance as futile. So you can see that from two sides. It's either we resist the parasite in developing more drugs and implementing all those measures, or the parasite resisting us by becoming resistant to drugs that are being developed or other measures that are put in place. So as you know, malaria is a deadly disease if not treated quickly. In 2021, 247 million cases were reported and 619,000 deaths. Amongst these, Africa carries by far the biggest burden of around 96%, as you see in this graph over the past 20 years. And death mainly occur amongst children under the age of 5. This obviously also creates a big socioeconomic burden, as people that are infected, they show symptoms of fever, shaking, chills, muscle aches, tiredness, and in advanced stages also anemia, because this is a blood stage disease. So obviously these people can't work, these people can't take part in daily life, and this is an added burden to the sickness itself. The parasite is transmitted by the female Anopheles mosquito, and to humans there are 5 Plasmodium species that are harmful. That's Plasmodium falciparum, which is by far the main parasite species prevalent in Africa. P. vivax, which also takes part in Africa, but is mainly dominant in South America. And then Malaria ovale in Knowlesi, who don't really play such a big role. There are other Plasmodium species that infect animals. Just one to mention is P. burgii, which is the parasite that can infect rodents. So what measures are taken to eradicate malaria, or what is important to put in place? First of all, you need research and development for new tools. As I mentioned, the parasite is transmitted by Anopheles mosquitoes. So vector control is one measure that one can take. There are insecticide-treated nets, as you see here on the right. So sleeping nets, as you may know, they obviously would kill the mosquito if they are treated with insecticides. Then there's indoor residual spraying that also uses insecticides. And this is done to help transmission, but these measures are also imperfect for eradication. One thing that people have observed, which is quite interesting, is that mosquito populations can actually adapt. So sometimes you see, instead of biting in the evening, and indoors they start biting outdoors and during the day. So that just shows that insecticides treated nets indoors are not a complete measure. Nonetheless, they still contribute to eradication. And South Africa has around a bigger than 85% coverage of indoor residual spraying, which helps control transmission. Another option is to develop better malaria vaccines. We'll briefly cover that later. New antimalarials, we'll cover that as well, and insecticides. That's not part of it, although I just want to highlight that the use of DDT was quite predominant like several decades ago, until it was banned, for example, in the US because of its harmfulness to human and additional wildlife. So one has to be very careful when developing insecticides that we don't harm ourselves in the end. In addition, new genetic technologies are being developed. So these can, for example, alter mosquitoes' ability to transmit the parasite. There are very interesting approaches out there, but these will also not be covered today. Then we need access to affordable, quality, people-centered healthcare and services. So this is obviously a big thing. You need a good infrastructure. You need diagnostic tools to diagnose malaria. But it's not only about having hospitals or community centers in place. You obviously need them in the right place. You need healthcare workers that can quickly go to the patient into rural areas. You need the right infrastructure for that. You may imagine that not everyone has cars or even bicycles. I've just recently seen that, for example, if people are treated in the Amazon, there are obviously villages that are only accessible by boat. So this puts a lot of strain also on the healthcare system, or on the ability to help. So these are all things that one needs to take into account. Yeah, quite recently, the WHO has recommended to add a low dose of primaquine to treatment options to suppress transmission. So far, it was only, I mean, the treatment obviously is the primary focus, that people are relieved of their symptoms. And the addition of primaquine, the transmission doesn't really have a benefit for them directly, but obviously it's to prevent the disease from spreading much further. So we hope that this will have an effect. But transmission blocking agents is a big topic as well. And we have very good scientists here in South Africa that actually look into that. For example, in Marie Burkholz at the University of Pretoria. Overall, this all requires money and international funding. So this is always a big topic. Then in addition, we need surveillance and response. So obviously we have to take into account all the trends around the globe that contribute to malaria spreading or the parasite spreading. You have to include policies into urban design. So for example, mosquitoes obviously grow in stagnant water. So these kind of things you can take into account, that you have as few sources as possible for mosquitoes to grow as a vector, and to stop transmission that way. Then yeah, malaria is obviously not a local problem. You can't say, okay, if I've eradicated malaria in one country, that's it, because you always rely on your neighbor to do the same, because mosquitoes unfortunately don't stop at the border. So transmission is a cross-border thing. So you need to cooperate with your neighboring countries. you need strategies not only at the national level, but also at a regional and very local level, because the demands may be very different depending on what your infrastructure looks like and what the people need in this case. Okay, so I want to briefly talk about the life cycle of the Plasmodium parasite, which is quite a complex one. It's actually, from a biological point of view, it's remarkable because pretty much every stage of its life cycle, the parasite has a different face. It has a different shape, it has different protein expression profiles. The surface looks different. So that will play a role if we look at the vaccines later on, because obviously vaccines rely on antigens that they can detect. So if the parasite looks very different at every stage, then you have to basically almost tailor the vaccine to a specific stage, or you do a mixture of different antigens. So I mentioned that the mosquito transmits the parasite, so that's where we start. It injects the parasite, so-called sporozoites, into the skin. From there, it gets into the bloodstream and into the liver. Then those sporozoites infect hepatocytes, so that's liver cells. In the case of Plasmodium vivax, there are so-called hypnozoites, which is a dormant form of the parasite that's very difficult to kill. So if you have Plasmodium vivax, it might be that 2-3 months later, after you're symptom-free, you think like everything is okay, and you get a relapse in malaria because all of a sudden the parasite awakens again and the life cycle starts anew. From there, from the liver cells, they spread into the bloodstream again and infect red blood cells, erythrocytes. And this is where the clinical symptoms start. So the so-called asexual blood stage cycle also has different forms: the ring form, the trophozoite form, and the schizon form. It's about 48 hours long. And every time these parasites, they burst, or the red blood cells burst. Obviously, red blood cells are being destroyed. Parasites multiply. The load of parasites increases. They infect new red blood cells, and the whole cycle starts again. So this is the part where the anemia comes in, because the body is deprived of red blood cells. This obviously has an impact also on transporting oxygen and so on. And at some point, when the patient or the host is so sick that it's about to die, there are obviously a lot of physiological signals that the parasite can pick up. And then it basically says, well, now it's time to get out. And that's when it starts to form gametocytes. There are 5 stages of gametocytes. The last form is sexual stages. And these are being taken up by the mosquito again. And then the parasite further develops in the mosquito gut first. So there are macrogametocytes, female ones, and the male ones. There's a process called exflagellation, where they get these little arms everywhere. And then eventually the ookinete starts to walk into the salivary glands, and it further develops into the sporozoites back again, and the whole cycle starts. So this basically gives us a few angles of attack from a drug discovery point of view. So first of all, we want to combat relapse. So that's for the hypnozoites, to kill hypnozoites. As I said, very difficult. Then primary focus always is to relieve the patient of clinical symptoms so that they feel better, which is that primary focus is always targeting the asexual blood stage cycle. And then eventually to eradicate malaria is to prevent the transmission back to the mosquito, because that's then, if you have a mosquito with parasites, that can infect other people, and so the disease spreads. So if we develop drugs, we first of all need a focus on what we want to achieve. So that's the target product profile. TPP1 is treating the patient. That's always the primary focus. TPP2 is chemoprotection or prophylaxis to prevent not the infection necessarily, but at least to the symptoms and to kill the parasite before it becomes a burden to the human. Then we have the target candidate profiles. They basically describe what properties we want the compound to have. So TCP1, that's, as I said, priority targeting the asexual blood stage cycle, which is responsible for the clinical symptoms. TCP3 is activity against hypnozoids, that's preventing relapse. As I mentioned as well, that's for P. vivax and P. ovale. Then we have TCP5 and TCP6. TCP5 is transmission blocking capabilities, so that's activity, for example, against gametocytes. And there's TCP6, which is targeting the mosquito vector. If we look at TPP2, chemoprotection, so far a lot of focus, like specifically from MMV, Medicines for Malaria Venture, who's like a huge organization. I mean, not huge from a personnel point of view, but they have a huge network and coordinate a lot of activities towards developing antimalarials. So far, the focus has been on TCP4, which is liver trizont activity to kill the parasite before it actually gets to the life cycle stages. But recently, the focus has shifted more to TCP1, long duration and fast killing agents, which would also help clear the parasite as quickly as possible from the blood. Obviously, you need a long duration for chemoprotection because you don't want to take a pill every day or even more often to prevent malaria. And TCP4 is still a good additive to a drug because it's basically a causal prophylaxis to prevent the clinical symptoms altogether. So what is being used at the moment globally? The primary focus or the primarily used artemisinin combination therapy is artemether lumefantrine with 75%. You see also the prices along the range there. They're quite cheap actually, and that is one focus. Obviously, if you develop a drug, in the end, it needs to be affordable because, as I mentioned before, malaria is a disease that is like predominantly active in Africa. So we can't have treatments that cost, I don't know, $50 or so per pill. On the other hand, I've mentioned that the highest burden is on children under the age of 5. So we need child-friendly formulations. We need to know whether it's actually suitable for infants, like very young children, or even safe to use for pregnant women, which is a big focus at the moment, also for MMV. It obviously needs to have regulatory approval. One big thing is it needs a low propensity to resistance. So obviously, we don't want a drug that after 1 or 2, 3 years of use cannot be used anymore because resistance has spread through the parasites. So these are all things that we need to look at. If we're looking at treatment specifically used in South Africa at the moment, as per NICD guidelines 2019, no surprise, first-line drug combination is artemether lumefantrine. For the chemists amongst you, I've put the structures up. always gets me quite excited. Then the second option, I was a little bit surprised about that, is quinine, actually in combination with one of the antibiotics, doxycycline or clindamycin. I assume that's because it's relatively affordable, although one has to keep in mind that there is resistance around for quinine and similar drugs. Then for non-falciparum infections, chloroquine or primaquine is used. Primaquine, I mentioned, is used as an addition to reduce transmission as well. So in addition to artemether lumefantrine. And when it comes to severe and cerebral malaria, then intravenous administration of artesunate at the bottom here is being used. In terms of prophylactics, we have atovaquone proguanil combination that's available amongst others under the name of Malarone. That's one that I've taken at least. I didn't have any problems with that. I know that for other people it's different. Usually it's mefloquine that is a little bit more known to be causing quite a lot of trouble in terms of some people have even reported hallucinations, obviously feeling unwell. So people usually are very careful with whether it's really worth taking prophylactics. But I mean, that's something that you should consult your doctor about. I mean, then there's also obviously malaria is a seasonal, at least in South Africa, where it coincides with the rain season. So there are times when it's safe to go without prophylactics. Then in addition to the South African recommendations, then the CDC, for example, in the US also has used chloroquine, which has also a very long half-life, primaquine, and tafenoquine, which is one of the few drugs that actually acts against hypnozoids. So if we look at the life cycle altogether, and you see that there are a lot of different drugs. There are different classes. They target different, different protein targets within the parasite. They target different life cycle stages, which in the end depends on whether the protein is expressed in that specific life cycle stage. This is just a general overview. I mean, highlighting things like artemether that we just heard about, in general endoperoxides, and other compounds like pyrimethamine. Those are DHFR inhibitors, and I will talk about that shortly as well. It's a very nice reference, definitely worthwhile reading. So obviously, with a lot of drugs, we have a lot of options for the parasite to generate resistance. What is drug resistance? It means that a compound shows weakened activity against the parasite. And this is usually, there's two different options that I will talk about in a bit. So emerging drug resistance is an ongoing problem. And that's why we need continuous development of new antimalarials. It's very interesting to see that most resistant parasite strains are actually first observed in Southeast Asia. So if you see on the right here, you have chloroquine that eventually was first observed, resistance was first observed at the Thai-Cambodian border. When sulfadoxin and pyrimethamine were introduced, resistance was observed within the first year of clinical use. So that's quite remarkable. And there are obviously reasons for that. The reason why it exactly happens in Southeast Asia is not entirely known. I mean, people assume that it may have to do something with that people don't comply. So they maybe stop taking drugs, the antimalarials, as soon as they feel better, so they don't complete their course, which means that there are still parasites left in the body, and they may start growing again. Obviously, if there is some drug pressure, they will generate resistance, and just by the sheer numbers, I mean, there are like several millions, billions of them in the body. So statistically, that's just a matter of time. or they might use lower doses than prescribed, which obviously then has a sublethal doses for the parasite. So that's an optimal breeding ground almost for resistance. Then, yeah, what kind of mechanisms of resistance do we have? So one option would be that you see a mutation in the target protein. So I've mentioned pyrimethamine, that pyrimethamine's target is DHFR, dihydrofolate reductase. There are also other options that you target a mechanism more. So in terms, for example, chloroquine is a hemosone inhibitor. I will also mention that in a bit. And the mechanism of resistance in that case is not a mutation in the target protein. It's more that you have a mutation in a transporter, and the transporter then more efficiently effluxes chloroquine in this case, or also related drugs. So resistant mutants can be also generated in vitro. So long before they're obviously on the market, we can assess whether it's very easy to generate resistance or if it's very difficult. So this is done with an assay where we determine the minimum inoculum of resistance. And this is obviously a piece of information that feeds into the drug development. So we want to have compounds that target protein targets that have a high MIR. So it's not easy to generate resistance, just to avoid this. And there are obviously other approaches. I mean, you've seen that in drug, the drugs that are being used are used in combination. So artemether and lumefantrine together, just in case you have resistance against one of the drugs, there's still another drug that can basically sweep up the rest of the parasites. And kind of clearing all the parasites and killing all the parasites just is the best option to avoid the spread or generation of resistance. So yeah, just to cover a few examples of drug targets, past and present. So one is the hemoglobin degradation pathway. I know it's not really a drug target per se, it's more a mechanism. Obviously, the parasite needs amino acids to survive, to build proteins, and so on. And this it gets from the degradation of hemoglobin. So that's why it's in the red blood cell. It uses plasmepsin and falciparines which are proteases to degrade hemoglobin to peptides. And what is left in hemoglobin, hemoglobin has a protein part and a heme part. So what's left is the free heme, and the free heme is toxic to the parasite. So the way it avoids being killed is that heme polymerizes into a non-toxic crystalline form, which is called hemosone. You see that here at the bottom. Drugs like chloroquine, they bind both to free heme and hemosone and basically prevent the crystal from growing, which leads to toxic free heme, which then in turn kills the parasite. It's also quite interesting to note that this all takes place in the digestive food vacuole. So the pH in here is a lot lower compared to the surrounding, which is why the basic center is actually quite crucial, because the activity or the binding itself is not that strong, but because of the basic center, chloroquine actually gets trapped and accumulates to higher concentrations, which are then obviously lethal. So often when you have in drug discovery basic centers and your basicity drives activity, it's quite interesting to look at whether it's actually a hemosone inhibitor. It's not a must, obviously. It doesn't have to be a hemosone inhibitor, but it always makes me curious. And then you see here the CRTs, the chloroquine resistance transporters, which are responsible for effluxing chloroquine in case of chloroquine-resistant strains. So another target would be dihydrofolate reductase, DHFR. I've mentioned that briefly. It is part of the folate metabolic pathway, which is needed to build nucleotide building blocks. And cycloguanol and pyrimethamine are antimalarials that target this enzyme. And it was mainly used after chloroquine resistance started to spread. I mean, as I mentioned, it didn't last very long until the first resistance was observed. But nonetheless, a drug that at least proguanol is still in use as part of the prophylactic. Something that's maybe interesting to mention is that proguanol is a prodrug. So in vivo, it then only cyclizes to form cycloguanol, which is the active metabolite. And you see this general motif here of the H-bond acceptor, H-bond donor, acceptor, and donor. You would see that here as well if I had drawn that in a different tautomeric form. Okay, so we get to developing drugs. So what you've seen until now is what has been used, what was developed, but I've also made clear that there is still a need to develop more drugs. And at H3D, we obviously have that as our primary target to develop drugs for the African patient. H3D is the first integrated drug discovery center on the African continent. We have biology in different disease areas. As you see, DMPK, in vitro and in vivo DMPK, and medicinal chemistry. And yeah, we are doing our best to develop new drugs. So just to briefly introduce you to the drug discovery process, H3D is part of the early drug discovery process. So we bring it up to a preclinical candidate. And we usually start with a screening. If you know your target, then great, you can screen against a specific enzyme target. If you don't know it, you can still do phenotypic screening, which in case of malaria means that you screen against the parasite itself. And if you have hits emerging from that, you would then start to do systematic changes to the molecule and see how activity changes, how solubility changes, cytotoxicity, metabolic stability. And there are obviously strategies for all of that to improve them. And it's an iterative process that you have to go through hundreds of times until you get like an early lead or a late lead ultimately. What I've shown here is a screening cascade. That means we don't do assays just randomly based on the target that we have. So if we want to develop an oral drug, obviously, ultimately, we need a drug that's permeable. So that's why we need something that's soluble. Only soluble drugs can permeate the gut walls and be absorbed into the body. It needs to be stable, metabolically stable. So that is one of the assessments. We obviously want to make sure that drugs are not toxic. Excuse me. But we obviously want to make sure that they're active and they're actually killing the parasite. So if we have assessed that in vitro systems, then we will ultimately move into in vivo systems. And mouse is one of our preferred animals to make those first assessments. Ultimately, if we're very happy with all these properties, we can move into the so-called NSG efficacy mouse model. That is a model that can be transfected with human blood and can therefore be infected with a human Plasmodium falciparum parasite. And then if we administer the dose, what we want to see is obviously clearance of the parasite from the blood. and that most effectively at low doses. Then ultimately we do safety profilings, we test for mutagenicity and off targets and so on, and want to have a risk assessment going forward for lead optimization. And if that's all green and good, or at least manageable, that we can address that later on, we move into lead optimization. So one of the success stories that we had so far was MMV-048, which came from H3D in collaboration with the Medicines for Malaria venture. And this was the first clinical candidate developed in Africa. And it was also the first clinically validated, or it was an inhibitor of Plasmodium falciparum phosphatidylinositol 4 kinase, or short PI4K. It's much less of a tongue twister. And that was the first clinically validated kinase target in Plasmodium. So 2 firsts for H3D, which is quite remarkable, given where H3D started from, like 2009. So obviously there's always a need to develop new drugs, to find new drug targets. And there's a consortium of 18 different labs across the world, mainly funded by the Bill and Melinda Gates Foundation, which is the Malaria Drug Accelerator, or short MALDA. It's a very interesting consortium, very different and diverse capabilities, state-of-the-art methodologies and labs. So really exciting. You can check out their paper or their website. It's amazing work that they do. And they obviously aim to improve and accelerate early drug discovery process by identifying new essential and druggable targets. So all of them is important, obviously. If you already have a drug target and you create resistance, you don't want to have another drug against the same target where you may observe cross-resistance. Essentiality is important because if a drug target isn't essential, that means you can't kill the parasite if you inhibit the target. And druggable just means that you need to be able to develop a drug against it, which is also not always possible. Then, what kind of drugs do we need? What lessons have we learned from the past? So obviously we want something that acts fast. We want to relieve the patient of clinical symptoms as quickly as possible. And for that, we have a look at killing kinetics. We also want something with long duration. That's not only for prophylaxis, but also for treatment. The ultimate goal would be to find a single dose cure, which means that you give the patient 1 tablet and it's done. So far you need multiple doses. And that's where the compliance issue comes in. So, some people, they want to go home. They can't stay in the hospital for a few days and wait until treatment is over. So they get the tablets and you have to rely that they take them until the end. But some people stop taking the antimalarials as soon as they feel better. I've mentioned the problem of relapse. So that's one thing. And we want drugs that obviously need low doses, because that's then better or cheaper to manufacture. Ultimately, the drug is going to be cheaper and more affordable. So yeah, that's a good target. We want something new. So efficacy against all known field resistance. New drug targets, it's one thing, but we also obviously want something that doesn't easily generate resistance. This is where the MIR, or minimum inoculum of resistance, came in, that I mentioned beforehand. And then also, I mentioned before, we need something that's cheap, child-friendly, and ideally also for pregnant women. And that's just because of the patient population that we mainly see in Africa. Obviously, there are new tools that are being used that were not available 20, 30 years ago. So also MMV in combination with partners have developed a machine learning model based on millions of results and compounds. They fed that into a model, and that can be used, is accessible online, so-called MIPE, Malaria Inhibition Prediction Tool. And this is a great starting point. H3D is looking into machine learning and artificial intelligence as well. We start using that, developing our own models. So we've started a new age of drug discovery. Then something else about predicting efficacy. So obviously you want to be confident that whatever data you generate, in the preclinical stages, so mainly in vitro and animal models, that they ultimately translate into human efficacy as well, which is not always that easy. I mean, I guess for malaria, it may be a little bit easier because it's a blood stage disease. So usually your drug gets into the bloodstream. It's basically where it's supposed to be, while in other diseases, it has to penetrate into tissues, so that makes things a lot more complicated. But nonetheless, it's not as easy as it sounds. So MMV, for example, has generated a tool that's called MMV Solar, and it uses all kinds of in vitro data like hepatocyte or microsomal stability. It uses activity, it corrects for different bindings, Albumax binding, plasma protein binding, and so on, and basically predicts a PK curve from that and predicts efficacy as well. So a lot of interesting and exciting tools out there. Then I've mentioned, MMV is one of the main players in malaria drug development, and they support a lot of efforts across the world. So here you just see MMV supported projects as of March last year. There's a lot in the pipeline, a lot of things that are very interesting and promising. One of the things is ganaplacide lumefantrine drug combination. Lumefantrine is already part of the artemethyl lumefantrine combination, but ganaplacide is a new one from Novartis, and they have decided to move it forward into phase 3 clinical trials now. So there's really a few things coming through. Obviously, attrition is always huge, so you need to start filling the pipeline with a lot of starting points. You'll see that H3D is also amongst the on the list of initiatives that are developing new drugs, as well as in one of our collaborations with Merck that are also at the moment in later stages, and hopefully will transition into the preclinical phase very soon. Then one last topic that I want to cover is, because it probably would have been a question, is the malaria vaccines. It's not really my area of expertise, but it was quite interesting reading up on that. So I obviously mentioned that the parasite itself changes very much through its life cycle. And that's why usually you would target a specific stage of the cycle. And what I want to focus on today is the pre-erythrocytic vaccines, PEV, or also anti-infection vaccines, those that you will find here at the lower right. Those obviously target the stages before it gets to the asexual blood stages. And ideally, if you are able to clear the parasite before they reach the bloodstream, that would be the best one. That's what's actually called anti-infection vaccines. So there are two approaches, basically. Either you can generate antibodies against surface antigens of sporozoites. These are the form of the parasite that are injected by the mosquito in order to clear them from the bloodstream or to block them from invading hepatocytes. The other approach would be to stimulate T-cell responses that attack infected hepatocytes themselves. So one of the most known, I guess, vaccines at the moment, I think there was a lot of announcements back then, is Mosquirix, which is from GSK. And that's so far the one that has progressed the furthest. It has been favorably reviewed by the European Medicines Agency and is recommended by the WHO. It contains, amongst others, a part of a recombinant protein that reflects the major surface antigen of sporozoites, which is called the circumsporozoid protein. That's actually a very commonly used approach in different vaccines. Then looking at the phase 3 clinical trials, we've seen that after 4 doses, clinical malaria episodes were reduced by 36% in young children. So, I mean, that's not altogether very exciting. If you think about that, you know, children are exposed to that, basically their whole life if they live in these malaria endemic areas. So that means you would need to take that vaccine quite often, and that's after 4 doses already. And they found that efficacy is strongly reduced over 18 months. So it would almost be necessary to probably take it like every year. So there are other approaches like the R21 matrix M vaccine that was developed by Oxford University. It also includes the CSP as antigen, but there are different combinations that they use in combination with a different adjuvant, and that shows 77% efficacy based on the phase 2b clinical trials in children, and even is as potent after 24 months. So that's quite remarkable. There are other approaches. I've also read about a whole sporozoid vaccine that's currently being developed. So it seems like overall they've taken up quite a bit in terms of developing malaria vaccines. So it's very interesting to watch. Finally, I just want to highlight a few of the collaborations that H3D has within South Africa in the malaria space. I just want to highlight Prof. Lynn Marie Burkholz. She is the SAST chair in sustainable malaria control. She does awesome work with the team. So I really recommend having a look at her publications and or her website. Then Prof. Lisette Kokemoor. She is a SAST chair in medical entomology and vector control. She also does an amazing job. We're fortunate to collaborate with them for several years. Have looked at all aspects like TCP5 transmission blocking and TCP6. Visited their labs, and I know it's probably a bit strange to say that, but one thing that has impressed me, and it's obviously not the most scientifically impressive part, but if you want to assess transmission blocking, you do that in an assay that's called the standard membrane feeding assay. And you have to dissect mosquitoes. So if you have in mind how small mosquitoes are, and then just imagine dissecting a mosquito and then count the oocysts that are remaining, that is very impressive. You need a steady hand for that. Last but not least, I'd also just want to highlight the MMV network. I mean, they have so many collaborators across the globe. They do fantastic work, and we're very fortunate to be one of their collaborators for over 10 years now. So thanks to MMV also for funding us and for allowing us to be part of the drug discovery journey. Last but not least, a few infos or a few suggestions for reading. What I can really recommend if you're more interested in malaria and like all aspects of it, also malaria biology, but also the policies and everything. Have a look at the Malaria EDX course from Harvard. It's really worth it. And it's really mind-blowing how interesting the parasite and the whole biology around it actually is. So with that, I want to thank everyone, specifically our funders in the malaria space, and obviously the whole H3D team that are gathered here in Kirstenbosch in one of our outings. So yeah, thank you very much.